Okay, so what does the data file of processed HiC data look like? Well, there are a variety of ways to record loci association or interaction data. From an analytical standpoint, there is the matter of whether the data is a raw count or a normalized value. A raw count is simply the number of chimeric read pairs that align to each particular pair of different loci, while a normalized value is the raw count after undergoing some sort of normalization and/or adjustment. From a technical standpoint, there is the matter of whether the data has been recorded using a coordinated list format or a matrix format. When the data is recorded using a coordinated list format, all information is recorded as a list, with each entry on the list consisting of two genomic coordinates and a raw count or normalized value corresponding to the association between those two genomic locations. Only non-zero values are recorded. An example of this can be seen on the right. The matrix format, on the other hand, records all association values, zeros included. With the matrix format, all information is recorded as a table with each row and each column corresponding to a particular locus, and each cell within the body of the table representing the interaction/association count/value between the loci which correspond to that cell's row and column. An example of this can be seen here. So we know how the data files work, but how is it visualized in a way which people can make sense of? Well, HiC data is usually visualized as a heat-mapped matrix. On the left, we have a heat map of the interloci interactions across the whole genome for mice. At this zoomed-out level of resolution, it doesn't offer us any biological insight, but it does behave the way we would expect. The almost solid red squares, which form a diagonal from the top left corner to the bottom right corner, corresponds to the intrachromosomal interactions, and the fact that loci have associated intrachromosomally much more often than interchromosomally is expected. We also see that the Y and X chromosomes appear to associate far less frequently than with other chromosomes, but that is actually an artifact of the normalization process, since there are 2 copies of the other chromosomes, but only 1 copy of each sex chromosome. On the right, however, we've zoomed into a small locus to show how a particular association pattern can be used to identify CTCF loops. Here on the left, we have the heat map of the intraloci interactions for a part of a single chromosome. It shows some regions with more long-distance associations and some regions with fewer, which was used as an indicator of how condensed that chromatin was. On the right, we have a different approach to visualizing HiC data. Since the square-type heat map is symmetrical along its main diagonal, half the heat map can be omitted, and the remaining triangle can be rotated 45 degrees so that the main diagonal now rests horizontally. This allows us to display HiC data alongside track-based data, as shown here. Anyway, that concludes the presentation. Slides and supporting documents can be downloaded from the links in the description below. My name is Angelo Chan, and I hope you enjoyed this video series.